Today I'll be talking about the isolation and use of bacteriophage to control bacterial spore forming growth in dairy processes. Now, I want to give a little bit of background on that, as this research actually started when I was an undergrad. I started as an undergrad doing some research for IMP, and that was also funded by Build Dairy, and we were looking at the different levels of spores throughout their milk fractionation process. And that kind of grew into this project, trying to find ways of controlling spore growth in that process. So here's my video. To start off, I just want to talk a little bit about bacteriophage. What are they? I know that we're familiar with them, or maybe we've heard the term phage, especially if you're in cheese making. But just to give a little bit of an overview, a typical bacteriophage is a virus that has a specific bacterial attachment. It's not infectious to humans, and they are everywhere. If you've seen them before in cheese, you know that in cheese making, you'll rotate your bacterial cultures in order to not get bacteriophage. Because even though bacteriophage have some good uses, they also have some things that can make them not desirable in processes, such as in cheese making, there's what's known as a dead vat, and that can occur in mainly any fermented dairy products where there's a single culture or even multiple cultures. But what the phage does is it goes in, attaches to the bacteria, and then injects its DNA into it and replicates with the bacteria, or it explodes the cell and creates more of itself to then go and continue its propagation, creating a product that is useless because you can't make your cheese out of it anymore. So some of the good applications of bacteriophage are from these companies, at least in food, Omnilytics, PhageGuard, and Intralinks. They use it to target specific pathogenic bacteria such as Listeria, Salmonella, and E. coli, and also to protect crops. There's also some uses of bacteriophage that have been investigated for use in humans, but that's not applications to food. That's more treatments of diseases and stuff. So bacteriophage targeting spore-forming bacteria. Why would we even think about that? Well, or why do we even care about spore-forming bacteria, I guess? It can survive the pasteurization process and then later germinate, causing safety and quality issues. And for dairy milk powders, that leads to markets that you can't reach because your spore count is too high, especially with infant formula. If you're trying to get low spore count powders in liquid milk, it can cause different curdling issues sooner than you think for spoilage. There's also late bloat defects that can be found in cheese, and basically the safety and quality of these milk products can be compromised because of spore-forming bacteria. Now, my hypothesis is that phage can be used in milk to reduce or eliminate the bacteria that are forming these spores. So these are the steps that I'm taking to test this hypothesis. The first step or objective is to identify the bacteria that's most prevalent in this process. We're mainly focusing on spore-forming bacteria found in milk powders, but this can also be applied to a wide variety of milk products. The second objective is to find phage that can target those specific bacteria. Because phage have such a unique ability to target specific bacteria, they won't, or they shouldn't interfere with like the milk fermentation processes, because we're not targeting those bacteria, we're targeting other types of bacteria that form spores. After that, we'll characterize the phage, make sure that they don't have any virulence genes or anything that could harm or transfer pathogenic or toxic genes into another bacteria. And then we'll test those in pasteurization to see if the phage will survive that pasteurization process. So in literature and what I've found so far with analyzing samples, the most prevalent bacteria that are found in powders that form spores are Bacillus licheniformis, Anoxibacillus flavothermus, and Bacillus cirrothermophilus. This is Bacillus licheniformis. If you've ever isolated spore-forming bacteria from milk powders, you've probably seen a colony like this. It's pretty prevalent and typical. It has like this mucousy top layer, but this is what I also found, and it collaborated with all the research that's already been done. Then I was also able to find phage for this bacteria. These phage, you can see, they're clearing these zones of bacteria, and that's what's desirable. Both of these different phage that I found, this is 10 to the 9th, a dilution of 10 to the 9th, and so we know that that's about the concentration that we had of phage. Now, the next objective, this is basically where I'm at right now, but continuing with the first two objectives as well, to continue getting more phage and more bacteria that we can target, is to sequence the phage isolated and submit it to GenBank so that we can use that in our publication, and then categorize the effectiveness of the phage against different bacterial isolates to see like the host range, because some phage will specifically target a specific strain of Bacillus licheniformis, for example, but it won't target anything else. We would prefer one that can target like 3 or 4 or 5 different strains of Bacillus licheniformis, or maybe even branch out a little bit into some other bacillus as well. And then for our fourth objective, we will need to prepare high titer lysates, which just means high concentrations of phage, to test in the parameters of pasteurization to see if those phage can survive pasteurization and theoretically be added to the bulk milk tank pre-pasteurization to continue their work, and then after pasteurization to continue working as well to decrease the bacteria. So